I request Mahalakshmi ma'am, please felicitate Mr. Sandeep Jagtap sir. Thank you ma'am. I request Dr. Tambe sir to give the welcome address and details of the guest. Today's guest. I request Principal sir to felicitate Patil sir. Thank you sir. I request sir to give the welcome address and details of the today's guest.
Sir, already we are behind the schedule, so with your permission, I will spare only 2 minutes to welcome you officially. Good morning to one and all. It's my privilege to welcome Amit Mudgi, who is the General Manager for West Region, Lab India. Then Sandeep Jagtap, who is from Nasik, but he is working with the Lab India and he is heading the office at Bangalore. And we have very strong association and legacy with this Lab India. Since 90s we are using the instruments and we are happy and I am really lucky to invite both the stalwarts from the industry who are catering the needs in not only pharma but in every aspect, every region where the instrumentation is required. So they are in the process chemistry also. They are providing the highly sophisticated instruments like NMR also. And as we were discussing in the cabin, so there is an opportunity for the students because they are offering those who are interested, so they are ready to take students for the training for all the sophisticated instruments. So it is especially for the PG students, it's an opportunity. So don't miss that opportunity. Be associated with these people so that will make you a career and that will give the opportunity also, placement opportunities. So without wasting time, sir, I welcome you to the campus to Mahatma Gandhi Vidyamandir Pharmacy College, which is established in 93, and recently the staff members, a very good team is there, hard worker, and in the accreditation we have received A+ grade. Not only the staff members, it is because of the students also, because they have performed well at every aspect, not only the academics, but in sports also they are doing well, in co-curricular activities also they are doing well, and that's why we could achieve. And we reached to this position that we could get this A+ grade in the NAT. So thank you sir, and hope there will be the demonstration also for the students for the latest updates in the instrumentation. And today they are demonstrating infrared spectrophotometer, what are the advancements, so that will be demonstrated in the session. Welcome sir, and thank you.
Thank you sir. I request Mrs. Yogita Thomre madam, please introduce our guest Mr. Sandeep Jagtap sir.
Thank you, ma'am. Good morning to all. It is my privilege to introduce our today's guest, Mr. Sandeep Jagtap. He is currently working as a business manager at Lab India Analytical Instrument Private Limited since 2018. Sir has completed his MS in chemistry from NIPER in 2011 and B Pharmacy from RDCOP, Pune in 2009. Sir is currently managing Karnataka team, which included achievements of sales targets per annum, also leading PAT India product sale, which includes managing sales, application and product positioning for spectroscopy products like NMR, FTIR dissolution, AAS and food products. He is also actively involved in customer feedback, new development, software upgrade, 21 CFR implementation and escalation management. He was awarded as Best Business Manager from 2020 to 2024 for surpassing annual targets. He also worked as a field application specialist at Agilent Technologies India Private Limited, Mumbai from 2016 to 2018, where he was involved in development method solutions for forensic on molecular spectroscopy, imaging like Curie 630, 640, 680, FTIR microscopy, FTIR imaging, and AAS. Formerly, he has worked as an application specialist in Lab India Analytical Instruments Private Limited, Mumbai from 2013 to 2016. He also worked as a technical specialist at Perkin Private Limited, Mumbai from 2011 to 2013. So, with this short introduction, I would like to invite Mr. Sandeep Jagtap sir to guide our student on the topic Insights of 21 CFR and Data Integrity.
Hello, good morning. So, how many students are from the chemistry, medicinal chemistry, and physics? Then I think pharmacology you will have, right? Pharmacology, and then QA, quality assurance. Okay. So, before going for 21 CFR, we'll start with the very basic thing. So, most of I think are the M.Pharm students. Like, or the final year?
Final year.
Oh, no issue. So I think they are aware about the FTIR and the UV spectroscopy, right? So FTIR, NMR, or it is in the next semester?
No,
all of
it will start.
it will start. IR is completed. Okay. Oh, no issue. So coming to the pharma industry, why the today's session is required? There are two things. First of all, you cannot have any driver which is not trained on the car, right? If you bought a new car, will you give that car to the driver who is not trained on the like a car? Checkout driver could happen gadi devu ka nahi, right? It will not be possible. So it's a very expensive car. Why you will give it to driver? We need the experienced driver. So same applicable to the any company. If they are hiring any person, they need to have a person who should be aware about the instrument. He should be aware about the industrial technicality. or it may be, let us say, I'm not saying like 100% you should be confident about the instrument. Certainly it's like a mobile app. Whatever you are operating, then day by day you will be very much familiar with it. But coming to the basic concept, what industry requires, that we are going to discuss. Let us consider why this 21 CFR is important. Anybody knows about the 21 CFR? From student, not from faculty. If it is wrong, that still it is okay. But it should be two-way communication. Otherwise, you will be sleeping and then I will be talking, that will not be good. Right? So, let us consider. I'll give an example. During adjuvant, I went for the FTR microscope training in Melbourne, and I had a headache and also the fever. We went to a pharmacy. There the pharmacist was saying you should give the prescription. Without that, we cannot give the medicine. But then we told like paracetamol is an OTC drug. You can give it. And I'm also a pharmacist. So okay, they said like you give the declaration that you show something like you are a pharmacist or something. Then we can give the paracetamol. So paracetamol, like a Dolo 56, you know the price of the Dolo 56, like 10 tablets, how much is the price in India? 10 rupees? Like 10 rupees you will get, I think, 6 tablets? Generic, right? Any paracetamol. It may be Dolo 56 or it may be anything. So we spend there 600 rupees for the 3 tablets. Right? Now, paracetamol, which is manufactured in India, which is a costing, I think Dolo 56 is manufactured by the Microlab. If you ask them the costing, the product cost, it's just 50 paisa. Because it's very much like a cimetidine or ranitidine if you take, GSK Nasik. Ranitidine is also they are manufacturing. I think 12 lakhs per annum their production is there. So hardly they are spending 50 rupees. And we are buying it in the Australia or the US market, it is for like a 600. So you can see what is the margin we have. Right? So 1 rupees tablet you are selling to the like let us say the 200 rupees, that means you have the profit margin. That's why every Indian company wants to sell their drug to US market or the export market. But suppose this US market, if you want to enter, they have some regulations. Like suppose you are buying any, let us say the milk products if you are buying. First you will see whether it is ISI certified, whether it is BIS certified. You will not buy blindly any food products or anything, right? Why? Because there are some regulations. Like in India, FSSAI has some regulations. So same, the US FDA, Food and Drug Administration, they have some rules. And it is also the indirect, they want to suppress the Indian growth also. What happened with the Ranbaxy, you might, I think sir also knows, Ranbaxy was #1 company. Ranbaxy was giving a lot of products, and when they were selling to the US, entire US is depending on the Ranbaxy. This was like a case. Suppose if the Ranbaxy sell, I don't want to, if they say I don't want to sell my product to the US will collapse. Same like I think in COVID also you might have seen, they were requesting for us hydroquinone, I think anti-malarial drug they were asking from the ENTAS, and then we chloroquine, and they said okay we'll supply. So same situation happened. So they wanted to control the Ranbaxy and Indian pharma market. So what they did, they introduced the US FDA. US FDA is having a regulation called 21 CFR. That is a code of federal regulation. Okay, all of you are going to use this one in industry. All pharma industries are like saying we need the 21 CFR system. So what happened to the Ranbaxy? They did some HPLC analysis. You know about HPLC. Suppose you have the paracetamol tablet, you need to pass it with the assays. QA people will also come to know that it should be between the 95 to 105%. If you are doing this assay, it should be in between the limit of the 95 to 105. If it is not falling, then it will be discarded. Also, if you have the drug like a 98%, like a paracetamol in tablet, or the API means active pharmaceutical ingredients. I am talking about only pharmaceutical ingredients, not with the adjuvants or the anything like the tablet. Suppose you have the paracetamol, 98% it is pure, but 2% what it is. That also we should know, right? If 2% if you are taking like nitrosamine detection, like also everybody is discussing, why nitrosamine is not good? Because it's anti-cancer. Sorry, it is a cancer, so that cancerous. So everybody wants to have the something should not be cancerous. And US people are very, very scared compared to the Indians. So 2% impurity, you have to dictate everything, LCMS, NMR, you have to give the particular data. If this is a 2% RS substance called as a relative substances, it should be there, and that you need to declare. Then only you can sell this API of paracetamol to the USA. Otherwise they are not interested. Please note, for 1 hour we are charging, I think 50 rupees for labor. They are charging for 25 US dollar. So manpower is also the limitation, but in India we have that. That's the advantage. But if you want to sell your product to them, they have the regulation. So now we were talking about the Ranbaxy. What Ranbaxy did all the analysis on the HPLC, but they were not documenting it. Means HPLC calibration was done or not, that they were not checking. The records were not given. Suppose you have a car, you want to go to the Delhi with the same car with the 80 speeds. What you will do first? You will check whether the car will be okay. First you will do the servicing, and then there should not be any problem for the long run, right? That you will do. So in HPLC there is a maintenance for every instrument. In your instruments also we have the like a maintenance AMs, they call annual maintenance contract, where the person will come, he will check the standard whether it is perfectly okay or not. Then they will do the analysis. But what Ranbaxy did, all HPLC and the GCMS, they were not calibrated. They did the analysis and they sold around 1,000 ton tablets to the USA, and it was on the coastal line. 21 CFR was not launched that time. But what happened, the person of the QC manager or the manager who was handling all operations of Ranbaxy, he took the data in the pen drive. Okay, all the data which were there, it was with him in the pen drive, and that data he given to the US authority. Under compromise, he had given that all data to the US people. Then US people came in the auditing of Ranbaxy, and then they realized there were major problem with the machines and the data, and they were having the documentation part also. So they rejected 1,000 ton of tablets of Ranbaxy which were about to enter in the US. Everything was thrown in the sea. Now, and Ranbaxy was almost closed. So the USA said, if you want to come to our particular country, you have to follow our rules. And if the rules if you are following, then only you will be able to sell. But again, we will do every year audit. That audit also should pass. That's why if you see, if anybody has worked in the industry, the most of the QC managers are very scared about the audits. Why? Because their product should not be rejected in the audit. If it is rejected, they will not be able to sell, and that will be the huge loss. You consider the paracetamol tablet 200, and if I am having tablet in 1 lakh number, you multiply how much is the profit for that. So that is a major problem. That's why 21 CFR came in the picture. Now how the 21 CFR is started? Let us say you have the practical of simultaneous Q analysis. Equation method is there, na? I think you have, right, in the analysis. Anybody has performed? For what purpose we are doing it? Simultaneous Q equation method. Paracetamol and ibuprofen, I think you have performed, right? What you did, there was a UV observance, 254 nanometer. You like the standards, your calibration plot you have plotted. Then you have the unknown sample. Then you got to know the concentration, and that if the combination of drug is there, that is ibuprofen or the paracetamol, both concentration you have to give. And once it is done, then you are saying, okay, using UV spectrophotometer, you have completed this quantification. That is called as a quantitative analysis. And if you are checking anything, that is called as a qualitative analysis. Right? There are two differences. Qualitative means what it is, quantitative how much. That's why we are saying UV spectrophotometer, not UV spectroscopy. UV spectrophotometer. Meter means measuring. Mass spectrophotometry we are saying sometimes. Spectrometry sometimes we are saying, because mass also can give the quantification if you are using the HRMS. But NMR spectroscopy we are saying. Why? The NMR is the identification. Right? IR spectroscopy you are saying, because you are just detecting the functional group and the what is the structure it is, like that. But Beer-Lambert's law can be applicable to the IR also and the NMR also. Nowadays many people are doing quantitative NMR also. So that is a different discussion. Coming to the 21 CFR, so suppose if you have done the UV analysis, the first you are keeping the black, then you are making it to 0. Now after that you need to keep the sample. suddenly you got a mobile call and you forgot to keep the cover of the sample. Instead you kept the blank only and you started the analysis. Now results will be 0 because the blank is already minus. Right? Why you are keeping the blank? Suppose you have the water in the sugar solution. You want to analyze only sugar. What you need to do? First you need to minus the water and then the sugar will come. So water we will be keeping as a blank and then you will do the analysis. So now you have done the mistake. You got the reading 0. Expected reading was a 0.8 absorbance. That was not coming. Now you did this mistake. Now you don't want to show it to your teacher. What you will do? You will delete the file. Because you have not done the right analysis, right? Now you delete the particular file, now your teacher has come and you have started the again same analysis. Now your teacher will not come to know that you have done the wrong analysis, right? You are hiding that data. So this was happening in the pharma industry also. If any product is failed, they were deleting the data. But nowadays, all pharma PCs are not having the option of delete. You cannot even the right and click, you cannot do the cut and paste also. If you see in the Lupin, if you go, you just ask them the PC, the right click, do you have the cut and paste? That option is also disabled. Why? You cannot delete any data, whatever you have recorded on the particular instrument. So, if your product is failing, let us say the paracetamol, you wanted to have like around, your AC was 95 to 105 and paracetamol is falling on 94. And you want to pass this product. What you will do, you will just adjust some little bit, like in UV if you want to pass, you just open little bit the chamber and then the absorbance will be increased, right? So if you done this particular thing, it will be catch. So this data is the actually manipulated data. This data will not be accepted. So 21 CFR is always based on the 4 things. One is the integrity. What is meant by integrity? Can any student can tell? What is meant by integrity? Very simplified term. Like your teacher was telling you, like I will just go, I have some work for half an hour, you just complete the assignment. Now, once the teacher is out, you are operating your mobile, you are not doing your assignment because nobody is watching you. But in front of your teacher, you are doing all the assignments. So when nobody is watching you, but still you are doing your job, that is called as integrity. So pharmaceutical analysts also, when they were not monitored by the QC manager, but still they will do the same procedure. That is called as the integrity. Irrefutability, that means your data should not be rejected by anybody. It may be the US authority or the FBI scientist or FBI investigator. You should be very much confident on your data. That should not be rejected by the anybody. So that is also one of the criteria. Secondly, whatever the data you have, it should be protected. Now, most of you know about the validation master plan, right? How many of you know the VMP? I think you have VMP, right? Few people will have VMP. Validation master plan, IQOQ. Anybody has heard IQOQ? What is the long form of IQ? These questions you will be faced in the industry also. Once you complete the MPharm, that time in industry they will ask you what is meant by VMP. VMP is a measure for any you can say from zero if you are starting any company, construction also, VMP has to be there. VMP means validation master plan. Validation master plan means whatever, let us say I am buying one car, XUV300, and they are saying XUV300, we are claiming that 120 km per hour can go. Now 120 km per hour, if it goes and if it is a new car, under that installation qualification means if I buy the car, I will ask that person, you do all the pre-checks, whether the car is in the proper conditions, sensors are working, belt and all the sensors are working or not. Then he will do the OQ. OQ means operational qualification. He will drive the car with 120. Okay, it should go 120 without any hiccups for 100 km. Let us say the 100 km test. That is called as a operational qualification. After that, PQ. That means performance qualification. Now suppose I want to drive this car in the Nasik roads. Can I use the 120 km speed? No, because the roads are not in that perspective, and also The traffic is also there. So what I will tell them, I want to analyze this car speed at the 60 kilometer with the conditions of the roads, with the traffic. But I will drive and you will sit here and you will watch me. That is called as a performance qualification, which will be done by the user. So same like a UV spectrophotometer, since why I am giving example of UV, because you are aware about UV spectrophotometer. Once you buy the UV spectrophotometer, you will ask to do the calibration by our engineer. How to do the calibration of UV? What is the method of UV calibration? I think most of you have given GPAT, right? So this question is also in the GPAT also. Huh? No, but this MCOM students also, they have applied for the GPAT, so already they have studied. Let us say like final year students should complete their study before GPAT. I think when we were there, the third year gate people used to, I think the MGV also many people qualified. I applied in 2008, that time I was in third year, gate 2008. And I think Inamdar madam, last career only we were doing. So my score was 99.59, all India rank was 109. And final year also I had given, but you have to cover those studies in the third year only, then you will have time. Otherwise in the February, I think your study is there, like for the like your GPAT, then you have the project coursework also, sessions also will be there. So you will not have time. So try to read this Najma Inamdar madam, this career communication book is there. You should minimum read around 5 to 6 times. Then 100% you will qualify GPAT, because all courses are, all subjects have been covered. Okay, let us talk about like what I was asking, what is the calibration? How to calibrate the UV? Okay, IR, I think this must be studied in the IR also. Like if you read Pavia, if you read Chem, if wires somehow, you must be 101%, because it's very simplified. That's why people are referring. So UV will be calibrated by the Holmium filter. Okay, it is as per the USP. Holmium perchlorate solution people are using, or Holmium filter people are using. So it has to be calibrated like a UV is proper basically. Next one is the FTR will be calibrated by the polystyrene film, because it is certified by the NIST. Some third party person is certifying this polystyrene is good, and then that will be kept in the FTR. Then it will be calibrated, and then you will come to know, okay, my machine is working properly. Then mass spectrometry, caffeine. Caffeine also, caffeine or the vanillin also you can use. You just spike and caffeine peaks, you will get to know about the mass spectrometry. And if whenever you want to calibrate, then 1% ethyl benzene you need to add, and 1% chloroform. So these are the both tests are given for the calibrations. So coming to the like a UV spectrophotometer, if you have calibrated it, our engineer will calibrate it again. Once you buy the instrument, you will say every 3 months we have to do the calibration. You might have, Sir Tambe sir might have told you like faculty, all instruments are calibrated before NAC, like any exhibitions, or if all instruments are calibrated, their reports are there. Why? Because your instruments are working proper. This is the indication by the calibration. Okay, when I have the car, some repairs have been happened. Let us say car make like I got failure in the car. All are Marathi, right? Major. So, but everybody understand Marathi, right? So if I use Marathi, it is okay for you, right? Or should I speak in Hindi? Because you should understand. English is not important. The thing is that you should understand the concepts. Okay? सर आता समजा आपल्याकडे एक कार आहे आणि कार बिघडली आणि तुम्ही सर्व्हिस सेंटरला दिली. सगळ्यात पहिले तुम्ही काय कराल सर्व्हिस सेंटर कडून कार आल्यानंतर? व्हेरी सिम्प्लिफाईड. टेस्ट ड्राईव्ह कराल. जिथे फेल्युअर आलेलं होतं तीच पहिली टेस्ट तुम्ही कराल की स्पीड एक्सेलेटर जात नव्हता किंवा क्लच फिक्स झाला होता किंवा गिअर पडत नव्हता. त्यावेळेस तुम्ही चेक कराल की झालं होतंय का नाही. The same thing is happening with any repair of the UV. So calibration means first of all you will do this test like whether the UV spectrophotometer is passing or not. Once it is calibrated, then you start using. So in the 21 CFR, it is a 21st chapter of Code of Federal Regulation. In that the 21 title is there. Under that Code of Federal Regulation, under that 50 titles are there. But the first of all, if it is a part 11, everybody is using Facebook, right? And Gmail also. Now from your mobile, since you are using all UPI and PhonePe, when you are mobile, if you are logging with the Facebook, and you are going in the, like let us say the Bangalore, and you are logging in your Facebook, another device. It asks you, should I log or it is you? It asks, right? And if you log, you will get one email. Somebody has logged in your account from this location or this IP address, right? So this is called as a audit trail. Why I am simplifying, you should understand, then we will go to the machine. So first, Facebook you have password and ID, right? After that, once you log, if somebody has used your password and ID, you will come to know about through the email. That is called as an audit trail. Audit trail means it will monitor everything on your page. When you log in, when you logged out, everything will come. In PC also we have the audit trail. Then, after that, you will have the security data. But I will give you the very example. Can you share your Facebook mobile password with anybody? No. Nobody will share. Right? Nowadays, the digital payments is very risky. So the same thing happening in the pharma. Ideally, you should not share your email ID and password of instrument access to anybody. It was happened in the Intas. Intas was in the, I think, liraglutide, I think they were, liraglutide is anti-obesity drug. Nowadays it is like anti-diabetic also, but anti-obesity also people are using. They were entering in the Intas, audit was there, and auditor was not announcing their visit. Suddenly they came. That was at least from the US FDA. So it is not at all, they will not reveal you when they are coming. They came in the morning 7 o'clock. First they checked the registers. Like they were having the biometric, all the people, they checked the register. That register, it was there in custody. They went to HPLC room. HPLC room, they saw that everything, data was perfectly fine. But that employee whose name was there in the report, he was logging. So that date and time is also coming. The reports were there. They checked like yesterday's data. Yesterday data, the same person name was there. The report was there. But yesterday that person was absent. And in the particular register, entry register, he was absent. That means he was not there in the company. Point number one. But his idea, it was used. म्हणजे त्याचा आयडिया आणि ते यूज केलं म्हणजे कोणी यूज केलं मित्राने केलं कोणीतरी युजरने की तू युजर यूज करून HPLC मध्ये रन अनालिसिस रन कर नाउ इट इज आल्सो 21 CFR entire audit was failed. So this is how it is important in the 21 CFR e-signature means your signature will not be compromised with anybody. Again this signature has to be changed every 90 days. 90 days म्हणजे स्पेशल कॅरेक्टर जसं सेम आपल्याकडे जीमेल मध्ये किंवा फेसबुक मध्ये असतं तसं स्पेशल कॅरेक्टर एक नंबर हवा त्याला प्रत्येक इन्स्ट्रुमेंटला and that ID will be unique for the unique person. Now you will say ki like suppose the ID is deleted or the password I forgot, who will reset? So let us say the Tamil sir is like the principal, so now you can consider he is a QC manager. Now under him the faculties will be there. Faculties, let us say the advanced user. Now advanced user under faculty, the students will be there, they are the common user. So sir will set up the one hierarchy. Advanced user, they can edit the method, they can do the analysis. But users, they will have to just click, they cannot change any method parameter. Like let us say the simultaneous queue analysis method, if you are doing, you want to do the analysis on 255 nanometer, but the method is saying 254. You cannot change as a user. Advanced user, they can change, but if they want to take the permission, they will have to contact to sir. Sir, we need this method change. Then sir will ask, what are the main loopholes in the current method? Why we should change 254 to 255? They have to justify. Then sir will write that signature and then he will upload in his documentation. We have changed this, and then that method will be changed. So in your any instrument, you will have the administrator, you will have the advanced user, QC manager is also there, advanced user is there, and normal users are there. So it will be applicable to you. But in terms of that, these users will have one ID, password, unique. And that password, if you want to change, you need to contact the administrator. Then administrator will ask you 3 questions. One time in the Lupin, what happened? That person did the analysis on the UV, but he forgot to keep it covered, and directly he started the analysis. Okay. Now this is happened. Actually, he has not manipulated the data, but it is a human error. Now, how to correct that error? So they say you put him on the training for 4 days. After completion of his training, he will have to give an exam on the same UV. and that pass certificate sir will have. So if auditor comes, if auditor asks, this was a mistake happened, what are the corrective actions you have taken? That corrective actions will be documented. So now, unfortunately, we don't have data integrity in our course, like we pharmacy. We are in the Bangalore also, the Karnataka also, we are talking to the people that we should include the 21 CFR in the, like the QA people, it is mandatory.
It is there in the syllabus.
But only data integrity.
No, 21 CFR.
21 CFR is
It
there.
is there in the syllabus.
So everybody knows about 21 CFR.
Not everybody, but the PG quality assurance.
PG quality assurance, but at least I think, how many quality assurance people are there? So you know 21 CFR, we didn't answer, that's why I was like completely zero, you're not aware about it. That's why I'm saying. So 21 CFR is very, very important for others also. If the people from medicinal chemistry, if you are doing any HPLC analysis, there also the 21 CFR will be required. People, how many people are from pharmaceutics? Yeah, so pharmaceutics and dissolutions if you are doing, now dissolution is also 21 CFR. So in the dissolution also, if you have not added the tablet and you have started the analysis, it will be also captured. Okay, so this is overall for everybody it is required. So validation master plan will be required for dissolution also, it will be required for QA also, it will be required for UV spectrophotometer also. Pharmacology, yes, they can little bit compromise, but if you move to the clinical trials, then again 21 CFR. Okay. So, and natural products, if you are into the Himalaya, now Himalaya is like a natural product type of the thing. But again, they are moving to the 21 CFR because everywhere the documentation is very, very important. So, coming to the, in the 21 CFR, can you change slide? These are the two things. 21, part 11 is the scope is specific to the electronic records. Now, what it says, all the data, all the context will be loaded. The same thing is happening here. Electronic record should be with the server. So why I am telling you, you might find it is a very irrelevant discussion, but after completion of your degree, you will face these questions. And when you are asking something in the interview, you should not be blank. At least you should be able to tell long form of treatment and what it involves. So selections, please note, when you are having a walk-in interview, it's very difficult to get the root package. Currently for M-Pharma, average package is 18,000 or like 20,000. And I don't want to demotivate, but you should know the reality. Now you calculate your fees and then multiply with the interest, then you come to know how much you are earning. If you want to earn good package, you should be very much well through about this knowledge. And then companies, when they came to your campus, you should be able to answer this question. If you hired by the campus, always the package will be. Let us say, X person started with the 4.5 lakhs per annum, let us say 35 to 40,000 per annum. After 1 year, he is switching to the next company. He will ask the hike 25% on based on the basic salary. 35% into 20%, around 45 for the 50,000 per annum. Suppose you have started with the 18,000 and you are asking 25%, how much it is? Maximum 25 or 27,000. जो gap है, that is compromise. So it is always better to improve this knowledge so that you can crack the interview. And it is, I have seen that communication certainly. Many of you are interacting with your people on the YouTubes and you are watching many movies, Hollywood movies. So if you practice English will come, it is not a one-day job, but that is not mandatory. Most of the interviewers I have seen, they are seeing your basic knowledge rather than your communication skills. Communication is secondary. If you are on the manager position, then communication will be improved. But the basic, if you are entering in company, that time you should know about the basics. Like a calibration of UV. This is a very simple question. If anybody comes, he will ask you for the calibration. Have you calibrated the machine? Have you asked to your faculties how to, if I am doing the project on the UV, I think many people will be doing the UV quantification one project. Have you asked like how to calibrate the UV? And if I want to do the calibration, what things I will use? So now I think, let us say that from today onwards, you should think about this. Okay. So electronic records already I explained. Electronic signature. The electronic signature means your name and the date will be there. So legally you are binding with all these documents. So in the 21 CFR, if you are doing validation, your sign will be there. If you are analyzing anything, sign will be there. Some people are saying manually sign also is required. That is also some people are practicing, but on the like a 21 CFR, people who are able to completely digitally transfer like a Biobond, this engine, MedReach, these people are asking directly e-sign records. Okay. So next slide. So if you see, Part 11 applies to all electronic records that fall under FDA, Food and Drug Administration guidelines. If an organization can prove to an auditor that their electronic record signatures are as trustworthy as paper records, ink signatures, the FDA will accept electronic instead of paper. This sentence is going to be made, which I told you. Some people are saying manually signs they are maintaining, but now the electronic records is also acceptable. The FDA will accept electronic submission instead of paper if those submissions added to Part 11 requirements are included among the type of documents that the FDA accepts electronically. So these are the detailed chapters on YouTube. You'll get the entire like a Part 1, Part 11, all the explanation in detail, but you should have time for that. Next one. Now, suppose you are facing the interview and you are completely blank about the 21 CFR. Just tell these 4 things. That person will come to know, like you know about the CFR. We just need to tell, sir, whatever the data I have generated, that data should have these 4 qualities. One is authenticity. Authenticity means I have done it, my e-signature footprints are there, my sign is there. Okay? Second one, I will share this slide to Tavis and sir will share with you. Okay? So first, like rather than writing, you should listen to me. You write. Otherwise, your entire things you will be writing the spelling of authenticity, everything, and then you will lose the topic. Okay? So in the slides, whatever I am giving, I will give to sir. Then you will have. I know after that you will not read for 15 days. If the 10% also will read, then that will be the success of these events. But I will suggest, sir, after every session, we should have the backup or some exam for the 10 questions. We will have 100%. I will also ask that. Then we will come to know whether we are better orator, better presenter. That also like a 360-degree type of the evaluation. Yes. So authenticity is important. Integrity. Integrity I have already mentioned. When nobody is watching you, you are following your discipline or not. That is called as integrity. Next one is confidentiality when appropriate. Confidentiality, very important. Now, let us take the example. Sitagliptin. Anybody knows radigliptin, sitagliptin? What is the pharmacological action of sitagliptin? Be loud, be loud. आणि ते जर गॅदरिंग असतं तर किती आवाज असतो तसा आवाज आला पाहिजे सीतागिल्टीचं काय ऍक्शन फार्माकोलॉजिकल ऍक्शन अँटी आय एम नॉट एबल टू हियर इट्स अ गिल्टिन इनहिबिटर yes or no sitagliptin ramigliptin okay anyway forget about it like let us say nimesulide or ketamine what is the action of ketamine I think sedative hypnotics झाला असेल फार्माकोलॉजी मध्ये आयदर बनवलं की मेडिसिन केमिस्ट्री झालं असेल so am I audible आवाज येतोय का last ला okay so if you have ketamine means it is like a let us say the sedative hypnotics under that so I want to prepare the ketamine as a new drug molecule. Currently, ketamine is there in the market, but when I am synthesizing, the isomers are having a racemization problem. That means ketamine मी synthesis करतोय, तर त्यातलं फक्त 50% मला active drug मिळतो. I want to increase the production. Let us say why I want to increase the production. Same ketamine method समजा मी जर 100 टन मला synthesize करायचं, same reaction. किती टन मिळेल मला? 50% current reaction आहे. 50 टन. Suppose if I increase the yield to the 85%, किती मिळेल मला? 85 tons. Means I am saving the particular money, right? So the same thing. If I want to do the new synthesis pattern for the ketamine, entire synthesis data will be confidential. Now, if you see any industry, if you are visiting to the industry, they will ask you whether you are carrying pen drive or mobile or not. Right? So nobody is allowed to carry the pen drive. Because that data should not be compromised. Because somebody has spent 10 years, and when that drug comes, then you will have to get the all amount. Why I think iPhone 18, why is it expensive? After 2 years, iPhone 18 becomes at half of the price they will reduce. Why? Because they want to recover the amount within 1 year, whatever they have invested. Right? The same applicable to the pharma industry. So if you have the atorvastatin or any drug, the cost of that drug, why the anti-cancer drugs are very expensive? Because still R&D is going on, and you need to recover the money. Once it is completely stabilized, the cancer treatment will be very cheaper. But right now it's expensive because many people are spending their money. So the same is happening. If the confidentiality is there, that time the electronic records people will not they will try to avoid it. They will use the papers. So your data should be confidential, but when appropriate. If you are into the ADM, then in the pharma there are 5 departments. One is R&D. R&D means it will do research and development. Then you have ADL. What is the long form of ADL? Analytical Development Lab. Okay, whatever the R&D has given the method, that will be validated by the ADL. If any loopholes will be there, suppose एक HPLC ची method आहे 15 मिनिटाची आणि त्याच्यामध्ये काही impurities अगर सेपरेट होत नाही तर ADL वाले परत R&D ला कॉन्टॅक्ट करतील की तुमची मेथड प्रॉपर नाहीये याच्यामध्ये हे करेक्शन्स करा. नंतर ADL ने एकदा ती पास केली ती मेथड ऑप्टिमाइज केली ती जाते QC कडे. QC मीन्स क्वालिटी कंट्रोल. म्हणजे समजा आपण 100 टॅबलेट्स दररोज मॅन्युफॅक्चर करतोय. 100 टॅबलेट्स चं क्वालिटी कंट्रोल पर डे झालंच पाहिजे. कधी कधी काय होतं की मॉटलिंग इफेक्ट होतात टॅबलेट्स मध्ये, कधी कधी कॅपिंग्स होतं. सो दॅट टाइम दॅट टॅबलेट शुड नॉट गो टू दी पेशंट. दॅट्स वाय द क्वालिटी कंट्रोल इज मॅन्डेटरी. सो क्वालिटी कंट्रोल लॅब ती मेथड अप्लाय करेल. नंतर क्वालिटी कंट्रोलने जे मेथड केलेली, जे अनालिसिस केलेलं ते क्वालिटी अश्युरन्सचं काम. टू सी द डेटा which is as per the 21 CFR, and assurance means you are assuring it. That's why it is a QA. Quality control means they will do the only test, but the quality assurance is a very risky job. Please note, if audit fails, many people from QA only losing the job because they are not I would say although they are doing the good job, but sometimes the mistakes that cannot be spared. That's why QA people will get a very good salary. You know, like the dahi handi, who is eating the makhan, who is on the top, but he will fall, he will die. आणि जे सगळ्यात खाली असतात ते त्यांना दही मिळणार नाही किंवा मक्खन मिळणार नाही, तर ते सेफ असतात. सो यू नीड टू कॅलिब्रेट युअर करियर ॲज पर लाईक दॅट. हू इज इन द टॉप, ही विल ईट द ऑल एव्हरीथिंग, शुगरी थिंग्स, बट द रिस्क इज ऑल्सो देयर. सो द क्वालिटी assurance is job like that. If you are qualifying any documentation, you should be sure. Otherwise, the first person who will lose the job after audit is the quality assurance. Okay, so quality assurance people should know about regular updates. What are the 21 CFR new updates are coming, and many people specifically for the QA, your career will be happening only quality assurance. And people, why this course was introduced? Because it was a very much need of industry. Quality assurance should be there. So scope is good, but if you are not ready, then that will be the problem. Okay, so confidentiality that we talked about, irrefutability, that means no way to deny that record is genuine. Means all the sample analysis will be the genuine. Okay. Now we will take the questions. Any questions from students? Don't hesitate. You need to ask. And if you are not asking now, that will be unanswered for next lifetime. So it will not be answered. So you have any question for 21 CFR? Either you are understanding everything or either you are not understanding. Like that is the only two way. Okay. So what is the long form of 21 CFR? We'll just revive it. Code of Federal Regulations. Okay. So which chapter it is? Which part? 11. Okay. Now suppose I'll just ask you one case study. You need to identify whether it is 21 CFR deviation or not, as per the 21 CFR. Okay. Suppose I got one sample from ADL. I analyzed entire tablet analysis I did, but there wasn't any peak for the tablet. म्हणजे समजा UV मी घेतलं त्याच्यामध्ये अनालिसिस करून तो एक पण पीक आली नाही UV मध्ये, ओके. आणि जे ड्रग्स त्यांनी जे दिलेले आहेत म्हणजे पॅरासिटामॉल जे दिलेले आहेत ते ॲक्युरेट आहे. ते म्हणाले की नाही हे रेग्युलर सॅम्पल आहे, पण तुम्हाला सॅम्पल रिझल्ट येत नाही, ओके. अँड नाव व्हॉट यू हॅव डन, यू हॅव स्टॉप द अनालिसिस, यू हॅव नॉट डिलीटेड फाईल, पण तुम्ही कंप्लेंट तुमच्या पीसी मॅनेजरकडे गेलेली आहे. इज इट 21 CFR or not 21 CFR? It is compliant or not? त्याच्या अगेन्स्ट आहे की कंप्लायंट? पीसी मॅनेजरला त्यांनी सांगितलं की पीक येत नाही मशीन मध्ये आणि सॅम्पल होत नाही अँड यू हॅव नॉट डिलीटेड द फाईल. इट्स अ राईट वे, राईट. नाव पीसी मॅनेजर विल टेल फर्स्ट की you contact to the engineer of that instrument because he is sure that the machine is right. Now, if we contact to the engineer and if the engineer says that no machine part has gone or the source has gone, if he is doing the repair, then this is compliant, 21 CFR. There is no any issue. But after the repair, what you should do? Should we start the direct analysis? What we should do? Right. So this is as per the 21 CFR. So you can do the calibration if they are understanding. So after that, you can move to the analysis. Okay. So this is related to the completely related to the 21 CFR. Now you have a PC which is separately given, and you have HPLC. आपण HPLC ने ऑपरेट करतो ना, HPLC ऑपरेट करताना PC ने करतो. आता HPLC is qualified, but PC is also has its own limitation. त्याला आपण म्हणतो CSV, Computer System Validation. Okay. So computer system म्हणजे कम्प्युटर मध्ये जेव्हा तुम्ही लॉग करताय, लॉग ऑफ करताय, हे सगळं कॅप्चर झालं पाहिजे. त्यामध्ये डिलीट हे ऑप्शन नको. इफ इट इज देयर देन दॅट इज अ बिग इशू. ओके. So if you are coming to the any record savings or something, your PC should be connected to the server. Server means all the data will go to the server, and that server will be completely secure. So your data will not lost for next 10-12 years. Okay. So this is the sub part, the electronic provision. But these 4 things you should know so that you can answer the question. Next slide, please. Organization that wish to use electronic signature must inform the FDA is writing prior to making the speech. Means some people are using now fingerprint, some people are using the passwords. If you want to make it speech, you need to write to FDA, and then if they approve, then only you can go. Each individual who will be using electronic signature must have their identity confirmed and unique. It will not be like Sandeep Jagtap, 3 people. No. Their email ID will be also different. Somebody will use Jagtap S, somebody will use Sandeep.Jagtap, somebody will use Sandeep.J. Like that. So their email ID and their names should not be same. Identity should be unique. For electronic signature, that make the user ID and the password passports, there are specific requirements for passwords and password generating devices. That means password when you are generating, it should be with a special character. Let us say that yes, A and D, at least yes should be capital. Then you need to add some number 2024, and then you will add the add or the some special character. That password only is acceptable in the 21 CFR. No normal password which is guessed by the others. Same like your bank payments, those people are saying you need to change the password. So this is also a part of it. Next slide, please. Okay, next one. So I am showing you the real-time 21 CFR on one resolution system. Okay. Just keep color correct. See, now this is the resolution system. If you see, it has an audit trail, it has the user ID, it has the user authentication, you have dual authentication, you have validation report, electronic signature, and data backup facility. Okay. Now one by one, you will come to know what I was explaining. Next slide, please. Now this is your PC or the like a resolution main unit where you need to have. If you are able to see, now let us say like one by one. Here's methods are there. Then you have the run. Then you have test report. Means first you will like set up the method. After method, then you will be running, and then test report will come. Function, where it will be used, very simple. Before starting the analysis, you will come to know whether the instrument is properly functioning or not. Okay? Then configuration, whether you want auto submit or not, that is the thing. And status means real-time status. Here you can see most of the people have used the dissolution. Dissolution is for the testing the like a tablet. This one. So this temperature should be as a body temperature. It should be 37. Okay. So now real time it is giving the monitoring of the temperature. Let us say like engineer mode. Any repair. I just told you, like if the instrument is not working, engineer will not use your ID. He will be having separate ID. So that is called as the engineer mode. So he will log in, he will do the repair, and then he will transfer to the user. Now you have the user management. User management means suppose, let us say that like you have the administrator, under him advanced user 7s are there, and then 7 users are there. If you want to increase the new joining, that time user management will do the creation of the new user and all. Now change password. As I told you, every 90 days you need to change the password. It will be done here. Audit trail, that already I explained what you are doing, that will come. Then backup with the server. And if you want to take with the USB data, that can be possible with it. So let us say, so this is what we were explaining, like method development. So any analysis will start with the method development. Okay. Then validations and all that we talk. So this is how audit trial looks like. Okay. Audit trial means by date, admin has logged in. Action, म्हणजे त्यांनी ऍक्शन काय काय केलं. जसं ऍडमिनने अगर समजा जर लॉग इन मशीन मध्ये केलेलं असेल तर तिथे ऍडमिनचं नाव येतं. इथे युजर पण असू शकतो. देन त्यांनी काय काय ऍक्टिव्हिटी केली इन्स्ट्रुमेंट मध्ये ते पिरियड, जसं लॉग आउट केलं किंवा एक युजर त्यांनी ऍड केला, त्याच्यामध्ये त्यांनी युजर अकाउंट एक लॉग इन केलं, परत एक कॉपी केली मेथड, ते सगळ्या ऍक्टिव्हिटी आहेत. नंतर डिस्क्रिप्शन, त्या डिस्क्रिप्शन मध्ये पण त्यांनी एक्झॅक्टली काय केलं, दॅट विल ऑल्सो विल द शॉप लाईक अ लॉग आउट, गिव्ह युजर ऍडेड, युजरचे डिटेल्स काय होते हे होईल, किंवा लॉग इन ऑपरेशन कॅन्सल्स, सपोज हँग झालं ते सुद्धा कॅप्चर होईल. नंतर न्यू मेथड सेव्ह, प्रिडिसोनेट, डिसोल्युशन कॅल्क्युलेशन हाऊ यू आर डुइंग, it's a pretty short time. Okay, so you will be calculating resolution with it. Now date and time, which date at what time, so everything will be in the audit trail. Some people wants to have the soft copy, some people wants to have the printouts. So this is also required for each and every machine. It may be UV, it may be FTI, it may be HPLC, it may be resolution. Okay, so this audit trail is common. Now everybody understand what is audit trail? Okay. Now detail of audit trail. एक audit trail वर आपण जेव्हा क्लिक करू त्यावेळेस पूर्ण डिस्क्रिप्शन मध्ये पूर्ण ते सांगतो की एक्झॅक्टली काय काय केलेलं होतं त्यांनी. ओल्ड व्हॅल्यू न्यू व्हॅल्यू मध्ये काही चेंज केलं का, डिलीट युजर केलं, येस इट शोज येस like that. So all the things with the date and time, everything will be there, because auditor will randomly they will pick up one audit trail and they will click and they will see this information. If it is wrong, if it is deleted without reason, the instrument is like that, data is compromised. Like that they will consider. So new method creations, you will be doing the method like this. It's very simple. Once it is created, you will give the right to the user. This user means, समजा जर युजरला जर हा राईट द्यायचा असेल तर हे टिक मार्क असेल, आणि जर हा नसेल द्यायचा तर हे टिक मार्क आपण काढून टाकतो. त्यालाच आपण म्हणतो युजर मॅनेजमेंट. ओके, म्हणजे लेट अस से ऍडमिनने लॉग इन केलेलं आहे, त्याला इथे जर युजर ऍड करायचं तर प्लस, याला जर युजर जर मायनस करायचं म्हणजे डिलीट करायचं असेल तर मायनस. तर त्यामध्ये काय की डिलीट, चेंज पासवर्ड, चेंज राईट्स, डिसेबल, डेटा बॅकअप, ऍड मेथड. समटाइम रॉंगली सपोज एखाद्या विद्यार्थ्याने डायरेक्टली मेथड डिलीट केली तर प्रॉब्लेम होऊ शकतो, म्हणून ते राईट्स काढून टाकायची इथे एक हे दिलेली असते फॅसिलिटी. So then delete method will be removed and the same the user management data will be moved. So user management means who has what rights, that will be defined under the user management. Okay.
So you can see the manager is there. Manager doesn't have these authorities. Disable users and configuration. These rights are not here. समजते परत repeat करू. Okay? So these are the things that user one इथे आता change झाले. इथे पहिला admin अमित नाव होतं. इथे user. He's a supervisor. Now supervisor ला फक्त काय आहे? Right? Database pattern. आणि इकडच्या delete method option काढून टाकले. Right? So वेगवेगळे पर्सनॅलिटी वेगवेगळे राईट्स असतात. Now user 2. User 2 म्हणजे डायरेक्टली स्टुडंट आहे. त्याचे राईटच आपण काढून टाकले. फक्त रन मेथड, प्रिंट अँड व्हॅलिडेशन एवढेच त्याला राईट असतात. बाकीचे तो काही करू शकत नाही. कळलं? सो दिस इज अगेन पार्ट्स ऑफ युजर मॅनेजमेंट इन द 2021 CFR. तर रन मेथड तर नॉर्मल आहे. तिथे तुम्ही डायरेक्टली ग्रीन क्लिक करून ते रन करू शकता. Now you will have the individual bowl temperature that you know the technicality. आणि त्यानंतर method approval. Now you have done the analysis, but supervisor authentication जे करायचं असतं ते authentication पण आपल्याला लागेल. म्हणजे 2-way authentication आपण म्हणतो. समजा लाईक सरांनी analysis समजा सरांकडे जर तो रिपोर्ट झाला नसेल त्याच्या अगोदर मॅडमकडे पहिला तर ते analysis ओके आहे का तुम्ही ॲज अ रिव्ह्यूअर तुम्ही ते चेक करू शकता. Once you say okay, then it will come to you. So it is a called 2-way authentication. So some pharma people are using. So like that, you need to write the comment here. Okay, the केलेले reasons जे आहेत ते ओके आहेत किंवा इथे जर तुम्ही रिजेक्ट करताय आन्सर त्यावेळेस तुम्ही सांगू शकता की this analysis is not falling in this under this circumstances. That's why I am rejecting this. So it will be there. But please note, everything should not be the past. People deliberately are documenting the negative results also to show the USFDA we have done the mistake. If somebody say we are completely perfect, not possible. All human will have some mistakes. So that has to be there. Again, that is also documented. So these comments will try to verify. Now the reviewer is seeing test is okay and review. Okay. And finally, approved release for use. Release for use म्हणजे सेल करण्यासाठी आपण आता डायरेक्टली मार्केटमध्ये टाकू शकतो. Okay, this is the approved release for use. Here, review that earlier slide. Here, quality assurance people will play the important role. If they want to review this data, they can do it. So, some industry people are using quality assurance, no other things. Got it? So, finally, it is authenticated by the final administrator, and the fault test data is okay. Now, validation. Validation, every 3-month validation should be there for the resolution. As per the most of the industry, they are doing performance qualification for any instrument every 3 months. So it is called as a performance qualification as per their own method. So some people are doing PVTA or some people are using the like a volume, temperature and the RPM. So volume, whether the withdrawal volume is 10 ml or it is 9.9 ml or it is 11.1 ml. That has to be validated. Then your temperature, whether the right temperature is coming in the bowl, like as per the USP, like a minus 0.4 degrees Celsius bowl to bowl, temperature should be there. So 37.5 and 37.6, like that range it should be there. So whether it is coming or not, RPM means how it is moving. That also you need to check. So this is the validation part. If it is UV, then I would suggest column filtrate wavelength accuracy test. If it is FTR, I would suggest polystyrene film. I will keep in the FTR and I will see whether the polystyrene, those are the 4 to 5 peaks are there. Anybody knows those peaks? These are also the questions in the entrance exam. Which are the 4 peaks for the polystyrene used in the IR calibrations? Like that. So those peaks are there, 4 peaks are there. Their plus minus is also given. If it is passing, it will move. Otherwise, no. Next slide, please. So it has checked the temperature also, how much temperature it is coming. Then next, I think it is checking RPM, real-time RPM moving. The next, then you will have the print report. In validation also, you need to have your signature and the signs. Okay. So after that, you will have this PDF. This PDF can be transferred to the the server, or it can be stored as it is as a system. So it is entire the 21 CFR operation.
basically. So whatever you have operated, now if you go for the any instrument, DISO will be a little bit easier for you because it's an integrated system. That's why I took the dissolution case. Now, next slide please. Okay. Now suppose this is a like a every system should have. Let us say you are coming next day you have some analysis. So if you want to achieve the temperature of your bowl 37 or everything, and if you want to set up the alarm, this feature is also there. Or next one, this function will be there. This is the case. Now first function system check. Suppose as per the 21 CFR you have come to the office and directly you have started the HPLC and you have started the analysis. Is it right or wrong? It's wrong because you need to see the pre-checks whether the instrument is working properly or not. So that's why the function system check is given. Whether the motor, lift motor, drop motor or the DG sensor are okay or not. If they are passing, then only you will do the next analysis. Got it? Otherwise, if something is happening, you will stop. Please note, some cancerous drugs are very expensive. Like around the one tablet is 2 lakh rupees. So if this analysis goes wrong, so you are wasting 2 lakhs of the company. So this should be completely avoided. For that purpose, the functions system check is given. The next one. So this is the configuration for the auto sampler. If you want to do the configuration for that, you can use. I think it's all into the configuration. Now you have any questions? You should have question, I think so, because after that only you will come to know like what is the 21 CFR. If you want to ask anything, VFP or the calibration, the PQ or the IQ, any questions? I think afternoon you will have the like a FTR this one demonstration, right? I think. Used for at least can you explain? I have slide, I will just open. So now we will have even USB 4 discussion. Let us say many of you are aware about the suture, ligature, medical devices. Let us say stents, right? Are they pharma product? Yes or no? Student, they are in the surgical, but there is a new term called medical devices. So now new year public, these are also starting with medical devices. I think NIPER has announced it. So medical devices testings, there is no any protocol, but now very soon it will come. So Mr. Amit Motki will take care of that USB 4, which is used in the medical devices.
So good afternoon. Myself Amit. I am working with this organization for 28 years. So I have been working with this company since it was in budding stage. So I have been involved in our product development. I have been involved in software, giving inputs to our software team, and of course, sales and marketing is my key responsibility. Since last 28 years, I have been handling that. So first, I would like to thank Tambe sir and the dignified members of this college for inviting us and giving us this opportunity, and to having interaction with you for this very key subject, as it was very nicely elaborated by Sandeep already. as far as the CFR compliance, which is going to be, in fact, it has been a very key factor in pharma industry since last maybe 5-6 years, or maybe I would say since 2017-18, it has started coming in, as he has illustrated a very great example of, not a good example, but very, say, the example of Ranbaxy, what has happened and why it has happened. So you should know each and every aspect of 21 CFR, and if you know it, your life is when you will go for your say career, that your life will be very easy. So that is why probably sir has hinted us to talk on this topic. So that has been already well explained by Sandeep. So practically, as he explained, authenticity, then total data integrity is the key factor. You should not have any loophole in your system or your documentation. That is the part, or that is the major part of 21 CFR. So we have recently developed another system which is again fully 21 CFR compliant system, that is USP4. As we just initiated the topic, that is sutures or stains, which are medical devices, which also contains a drug portion in it, and it is not directly consumed by a human or by a person, but it is being placed in your body's system through some procedure, and that is how your drug or the drug from that device is getting reacted for the respective cause. It could be your heart trouble, it could be your blood thinning, or whatever, your pain for some specific organ. So that is being for to cure, to have some say positive action for those problems. We are developing, or pharmaceutical industries have started working on this medical devices. So this USP4, that is called as a flow-through cell system. In this flow-through cell system, this is typically used. Of course, most of you are very much aware of basic dissolution system, where we have a limited volume. Usually, what is the volume you take maximum volume in dissolution system? Can anyone tell me? What is the bowel capacity in dissolution system? 1 liter. Okay. Of course, you get an option for 2 liter and 4 liter as well in dissolution. But 4 liter is the maximum volume you can adopt or you can use in dissolution system. So that is the maximum limit. But most of the protocols so far said for all the USP protocols or for all the pharma drugs are with respect to either 1 liter or 2 liter. But people are doing some researches with 4 liter volume also. But it has not been officially published with 4 liter volume so far for any drug. But yes, 1 liter or 500 ml is very commonly used, 750 liters is very common, or even 900 ml. Am I audible to the last benches? Okay. So this is the fact, and so in normal dissolution system you have a limited volume to work with. So many times when now people are developing more, say, or going with more intense R&D, they are trying to develop higher dosages for some reason. Of course, it's as per the demand of the medical treatment requirements. So people are developing 1000 mg drugs, 1500 mg drugs, 1200 mg drugs, which are probably not able to get the correct dissolution profile with this limited volume. So when such constraints are being observed, that is why this USP4 has been launched, where you have the flexibility to work with unlimited volume to check the drug profile. You can work with say 20 liters of volume, you can work with 40 liters of volume, you can work with 80 liters of volume. Even for sampling, you can withdraw there are two versions or two more, configurations. There is mentioned open loop and closed loop configuration. Okay, so in this open loop, you can collect as much as 80 ml of sample in particular interval. So your volume also is being increased for sampling, and your total drug profile, media volume also has been increased to a great extent, almost 10 times or maybe 20 times. If you go with say 20 liters of volume, so it is 20 times. Even you can go with 40 liters of volume. You can go up to 80 liters. So 80 times has been increased. The media volume can be increased with the help of this USP4 apparatus, where you are likely to get the better resolution profile. Or especially in case of these medical devices, you do not have any protocol or any method specified with regards to normal resolution. That is apparatus 1 and 2. That is basket and pattern. You do not have any method profile or mention about this medical devices study. Whereas with the new studies, with this flow-through cell, you have the protocol for stents, you have the protocol for patches, skin patches, with respect to this flow-through cell. So that is why nowadays flow-through cell system is in demand, and that is why we thought of developing this, because there is only one competition as far as Indian manufacturer is concerned, but they are still struggling, whereas we have, with proud I can tell you, with great success we have launched this system with no flaws, or in fact I would say we have much better flexibility, we have much better features, we have much better specifications even against the imported systems or imported brands in this category of instruments. So that is why very recently we have installed the first system at Lupin R&D Center in Pune, and with that experience of last 4 months or 6 months I would say, I think we installed it somewhere in May or June of this 2024. So with the experience of last 6 months, they have decided to repeat the same system at their Pithampur unit. Although they have earlier 2 systems at the same location, that is at Pune, they have 2 more systems from earlier bought systems from the imported manufacturer. The third, when they thought of buying a third system, they thought of giving a chance to Lab India, and that has been successfully accepted, and we have delivered the system successfully, and we are getting the repeat order. So that is the key factor. They also evaluated the other brand, which is the Indian make, but they couldn't get the satisfied results with that system. So we got the another chance, and we could prove that our system is passing or through with the imported system. So that is why practically this has got a demand nowadays. So next one. So this is a kind of in vitro or in vivo. Of course, you must be aware of these terminologies. In vivo is what happens actually in your human, body conditions, and in vitro is external simulation of the body conditions. You are creating the similar conditions with the device, especially if I have to mimic or give you the example in dissolution. In body, as Sandeep explained, our body temperature is 37 degrees. So we are maintaining 37 degrees as a bowel temperature. We have various acid, gastric acid, or various liquids, or so that we are using as 0.1 normal HCl or phosphate buffer in dissolution as a media to have the resolution profile. Same way we have some churning moments, the continuous movement in our organs within the stomach or intestine. It is contraction, relaxation. So that we are creating with the paddle. It is continuously having the movement. So this is a kind of simulation. Same way in flow through cell, as we have a blood stream, because these medical devices are being placed very close to your bloodstream, or when you are placing any patch either on your shoulder or on your back, you are placing it very close to your blood veins, which are flowing just beneath the skin part. So that is how this USP4, the cell which is shown in the earlier slide, this. You can see the cell. So in this cell, the media comes from the bottom and it goes through the drug, the yellow portion what you are saying. The yellow portion what you are able to see, that is your drug portion, or that is your drug part. And that has been kept in one kind of folder. So same way we can divide or provide various different holders for various different sample types. Is it audible? So that is how the media comes from the bottom and it goes through the sample and comes out from the top. And that is how you can keep your drug in continuous contact with the flow of the media. So your media is continuously flowing through the drug with a very specific flow rate. It could be say 10 ml per minute, it could be 5 ml per minute, 2 ml per minute. You can even set 1 ml per minute. The 1 ml will be going with a rate of 1 minute, or even you can set even lesser than that, 0.5 ml per minute. So that is why your drug will be in contact with the specific volume of your media for a certain time. And that is how you will keep on getting this slow drug release with a larger volume of media. And that is why with the larger volume, you are expected to get a proper profile in such cases. Either the drugs are not properly miscible in a limited volume, as I mentioned in dissolution, that is the maximum volume what we use is 2 liter, 1 liter or 2 liter. So whereas here you have a option of going with a higher volume of media, which will tend to give you better resolution profile. Okay. So this is a kind of profile what we get in vivo and in vitro with this apparatus 4. So these are a kind of different dosages, normal tablets which are not giving you the desired profile with the limited volume. You can work with this USP4 apparatus. If you are trying to perform the API profiles with the limited volume, and again if you are failing in getting the desired profile with the limited volume, you can try with USP4. The capsules palette, again if you face any insolubility issue with this USP 1 and 2 in normal dissolution system, you can go with USP 4 system, which will give you appropriate dissolution profile. Medical devices, as I mentioned, it could be drug-eluting beads, it could be stents, implants, coated drugs, or implant lenses, your corneal lenses, which are medicated nowadays for certain, before surgery, doctor puts those lenses or asks patient to apply those lenses for certain time so that the drug will be diffused through the corneal portion, and then it will be easy for doctor to go for the surgery. Those kind of devices have been developed. So those devices also you can check for the corneal studies with this USP4. suppositories or soft gelatin capsules, which is again a very tricky sample to get the proper profile with the regular dissolution studies. So that you can very well get the profile, good profile with USP4 apparatus. And again the parenteral emulsions, suspensions, where you face some problems with for getting a proper drug profile in normal dissolution basket or paddle. You can very well try this with this USP4. And this has been already proved, and that is why the system has been developed and it is in demand. Okay, the typical parameters here, what we said is dissolution volume, dissolution concentration, then agitation. Of course, this here the agitation is your media flow. There is no stirrer or anything placed in the system. The media flow itself is an agitation, and that is why your drug is getting released with a typical pattern. Then configurations, as I mentioned, there are two configurations, open loop and closed loop. And dissolution profile is the final data what we produce with this study. So if this is a comparison what has been illustrated in USP 1 and 2, that is our normal dissolution system, we have constant volume, that is 100 ml to 1 liter. Of course, this has been illustrated with 1 liter maximum capacity. So this is a limitation. Whereas in closed loop, you have a flexibility to work with 15 ml to 4 liters of volume. And in open loop, it is dynamic. As I mentioned, you can go as high as up to 80 liters of volume or whatever volume you want to work with as a R&D tool. Then this is dissolution concentration is cumulative. There is also cumulative, whereas there is a differential drug concentration as you are going to use the infinite media. So your drug concentration will be differential, whereas here in both these cases, since although it's higher volume, the volume limitation has been there. So you will have a cumulative drug concentration, whatever profile you are getting. Whereas there you are going to use, or you can use the infinite volume. You can pass the huge media volume through the drug. Your drug concentration could be differential. Okay, then agitation, here you are creating the agitation with the basket and the stirrer, whereas here, as I mentioned, it is liquid flow itself, which is creating some movement in the media. Okay, then evaporation losses here can be limited by using the closed design instrument. There also the losses are very much limited because it's a very closed loop system. And here also no evaporation, again it's being a closed loop system. Then configurations, as I mentioned, here you have basket and paddle, whereas here it is closed loop, and there it is infinite volume with the open loop. Dissolution profile, you can see, that is why it is cumulative, here also cumulative, and there is differential. That is, it starts, it is increased, and it is going to zero again, back. That is why it is called as a differential. This is what is our system. So, this is an open loop system where you have the main dissolution apparatus with 7 cells connected sequentially for placing 7 different drugs. As we normally see, if you have worked with 8 bowl dissolution, you have a requirement of keeping 6 tablets of same batch in dissolution, whereas here normally the practice is keeping either 6 or 7 tablets or your 7 drug devices in this 7 cells, and then with this piston form there is a media selector you can work with 5 different medias even for this drug profile. You can change the different medias after certain intervals. Like first, if you want to work with 0.1 normal HCl, if second interval you want to increase the concentration of HCl, maybe the third could be phosphate buffer. As when we discuss this with different scientists, they tell us that in esophagus there is one typical media. In your intestine there is a different media. In your stomach there is a different media. That is why these media changes are required. As you suppose I am taking some tablet for some reason, I am having some fever or maybe I am having some chronic diseases, for example. So in those cases, when I am taking a tablet, my tablet will be, say, for a certain period it will be passing through the esophagus. For a certain period it will be stationary in my, say, either small intestine or big intestine, or it could be a stationary for a certain period in stomach. So likewise, as we have different, say, secreting media in our body system, same way we can have the option of changing this media after certain intervals. So this media will be transferred to this piston pump and it will be circulated through these cells, and then ultimately it will be collected as a sample, certain portion. If you are, say, flowing 4 liters or 8 liters of media, certain portion after certain interval, it will keep getting collected in this sample collector. That is the last portion. And this is a media, 3 medias plus water. We can keep these containers. So open loop, this I will see because this is very theoretical. And this is again a closed loop system. As a advantage, as I mentioned, the flexibility with the small and large volumes. Both the small and large volumes of dissolution media can be used without changing the hydrodynamic conditions experienced by the dosage. That is from 15 ml to 400 ml, that is 4 liters. The ideal, particularly for low dosage drugs, forms in any form like solid, liquid or solids. And this is a system what we have for the closed loop system. In closed loop you get 2 pumps, one is piston pump, another one is a syringe pump. Because we have to have a limited volume which transfer the media from here, then it goes to the syringe pump as a sample collector, and then it is collected here. This is again the similar profile what has been shown in the earlier slides. So here, as I mentioned, there are various different designs for these cells what we can provide. So you can see the advantages. There is no floating on or sticking of this media. In normal dissolution, you can even have some problems like when you are dropping your tablets, it is sticking in some odd position on the wall of the bowl. So if it is get stuck on the wall of the bowl, it is very difficult to get the appropriate drug profile. Whereas here your drug position is always fixed. It is not going to have any variation in your drug profile because of its exact position. So that is why no floating, no sticking. So you tend to get a proper drug profile. Powder API is not compressed. You don't need to create a pallet when you are going with this USP4. Usually in dissolution 1 and 2, you have to have a pallet form or a tablet form. Whereas here you can directly place the powder. So that is an advantage. Okay, this is another design of the cell. which can be used for various drugs, cell for suppositories, soft gel, cell for powder granules, cell we can develop for implants. So, as I mentioned, various designs are available with us. This is for suppository. You can see the suppository tablet has been placed in one compartment. The sample will be coming in from the bottom, the media flow. It will be flowing through, and from the top, it will be getting down through the next compartment, and it will be collected. Likewise, the closed loop system can be designed. This is for stent. So this is a typical drug profile what has been studied with this USP4 for the stents. Okay, this is for topical products, semi-solids. Topical, it could be your skin patches or cream ointments. So this is designed for topical products, can be used as an alternative for an extraction cell or for the Francel. The Francel division, it could be an alternative to Francel study also. Okay, for a volume of up to 1.2 liters of a product. Yeah, here if you are working with a membrane, you have a two-way conditioning also. You can place in reverse way also, you can place in the say appropriate position also. Both way, the membrane facing direct to the media also is possible. Membrane facing opposite to the media flow direction. Both way you can study here. Okay. So this is smart development. What we call it as smart development is when the critical information is obtained earlier with the resolution being a supra indicator. The powder cell can be used for an apparent resolution as well as for the granulates and intermediates and the final dosage. So in any stage, this USP4 can be used. The granular stage, the API stage, or the final dosage form. Every kind of product can be studied with this USP4. So that is why it is called as a smart device. Qualification, it's quite similar what we had with this dissolution. We have to validate the flow. Of course, in regular dissolution, you have various parameters to be validated. Like you have to validate the RPM, you have to validate the centering of the shaft, you have to validate the depth from the bottom of the bowl. Whereas here, the only validating criteria is your temperature, cell temperature, and the flow. Only two parameters has to be validated in USP4. Whereas there you have at least 5-6 parameters, that is wobble, your depth, your centering of the device, your temperature, and RPM. At least 5 parameters you have to validate. Of course, there are additional parameters like coplanarity of the device that can be studied in normal resolution by using the digital protractor. Coplanarity is the parallelity with respect to the platform where you have kept the system. It should not be a tilted way. If it is tilted on the front side, back side, or sidewise, also you will get a variation in your profile, especially in dissolution, normal dissolution. Wherein USP4 has got no impact with respect to leveling or no impact, there is no RPM or no stirring device, so you don't have to set any RPM in this. There is only two parameters, is temperature and the flow. That's it. Okay, this system is called as a F7 Smart. This flow through 7 cells, So it is called as a flow through 7. F stands for flow through and 7 stands for the number of cells. And these are some of the features just for your say reference. You can go through 7-inch touch screen, then 6+1 test units. As I mentioned, normally 6 dosages and 1 for blank, or 7 dosages, or 6 dosages, 1 API or excipient. Likewise also you can go for the dissolution profile. Then individual cell temperature monitoring is there. You can set temperature from 20 to 55 degree. Even in our dissolution now we can have the provision if you for any reason in industry or anyone approaches you whether we can perform the study in dissolution, normal dissolution with paddle and basket. We have a provision to work with 10 degree media also, 10 degree temperature media. We have very recently developed this device for Cipla. They had one typical application to work with 10 degree for certain drugs. So that we have designed. Same way we can, in this system as well as in our normal dissolution, you can go up to 55 degree high temperature studies also. So that is also possible. So in our normal dissolution, now we have changed our specification from 10 degree to 55 degree. So that is an advantage with our dissolution. It has built-in validation program for temperature and flow, indication for cell blockage, because there is a possibility because the capillary which flows your media from bottom to top, there is a very small capillary kind of portion through which your media flows. So there is likely chance that if your drug or if you are working with say powders, if your powder flows down in the channel, your channel may get blocked. So system has the indication or has a sensor to sense whether the channel which flows the media has been blocked. If it is blocked, system will give you the indication or give you the error indication on the display that channel is blocked. If the cell is not placed properly in the position, then also system will recognize the position of the cell and it will give you the command or instruction on the display. If your temperature is not within the set criteria, then also you will get the instruction or get the signal on the display. So there is no possibility we have kept that user will create any error from his end as far as the system operations are concerned. Everything is controlled and monitored by the system with the advanced sensor-based technology. Okay, then vibration-free external temperature controller. We have unlimited method for analysis. The device has got an infinite memory, so you can set number of methods as you want to develop. It could be 100 methods, 200 methods, 500 methods. Same way for data storage, we have unlimited data storage. Of maximum we can give you 32 GB memory. So at ease you can store your data for next 5 to 6 years, even if you are using this system daily. The same feature is available with our resolution also. The data memory, the touchscreen which Sandeep showed you, that has got an infinite memory. So the earlier version, the LCD display has got a limited memory. You have 15 programs, only one result will be stored. Wherein the touchscreen device has got an infinite memory for all these things. It has got USB, Wi-Fi connectivity. You can connect a Wi-Fi based printer also. You can connect the system to LAN connectivity. You can store your data to your local server. All those features are there. Because all these features are very much required when we sell this system to any pharma company. Nowadays, they do not accept any system which is not LAN connected, which cannot be a LAN connected device, because they even test tubes in a row. Likewise, you can collect 13 sample intervals, wherein closed loop we have limited volume collection. So with this 7 test tubes in a row, Likewise, you can go for 24 collections in same profile. The volume collection for sampling could be 1 ml to 80 ml, which I explained you in open loop device. In HPLC oil tray, you can collect this same volume in HPLC oil directly also. So that is also possible. So in closed loop, you can go for 15 ml test tube, which can withdraw 2 ml to 10 ml for the closed loop. Then automatic sample collection as well as waste collection is also there. So in between two profiles or two collections, if you want to clean the entire sampling tube part, you can give the rinse functionality, you can use the rinse functionality. So before your next sample getting collected, system will ensure that nothing is left for the earlier sample portion before the next sampling is being taken. This is available in this flow-through cell as well as in your dissolution. So you are always ensured with this rinse functionality. Your next sample is collected without any contamination or any addition of your earlier additive. So you are not getting any contamination issues, neither you are getting any dilution effect with this function. So color indication for auto manual sampling, ready condition in single multicolor LED. And media selector, which is already I showed you. 5-port media selector, automated media change for 3 different medias, indicated port for water as cleaning media. So which I explained in rinse functionality, you can go with the water cleaning. In waste collection port for clean cycle. In colored connector, again to identify which media is going to which part. This is a complete picture for so that is all for USB 4. So if you have any queries, any doubts, anything, you can either ask now or you can approach sir. He can approach us with mail or you can have our mail also. So with this, again, I would like to thank Tambe sir and all dignified college faculties for giving us this opportunity. So thanks for this. And the next session, we will have the hands-on with our FTIR. We are associated with Thermo Fisher for their entire FTIR segment. We are also promoting their Raman and FTNIR systems. So in afternoon session, we will have the hands-on demo. We have brought this system. So you will have the demo for the FTIR. It is not yet open. Sir was trying to see. Today आपण उघडू दे and we will give you the thorough demonstration on that. It's a very beautiful system. So त्याचे features वगैरे आपण second half मध्ये आपण explain. Okay, thank you.
Thank you, sir. I request Akade sir